whole time, the whole thing was shadowing us, right behind us, right on the side of us. You could kind of see the thing moving through the woods.
All I can remember is flipping the light on, and I see this creature, and I knew in my heart, I knew in my mind, the whole night, this isn't a man.
And then this thing walks across the road, takes a turn towards us, and then leaps over a guardrail.
Went to look forward, and there was a big black thing, is all I can call it.
Welcome to Squatch-D TV, exploring the Bigfoot mystery each week with your hosts, veteran researcher, author, and TV personality, the Squatch detective, Steve Kulls, and from the Bigfoot Research Project of Kentucky, Chris Bennett. Sit back and buckle up as we bring you guests from around North America discussing the Bigfoot phenomena, but not without a few laughs too. Here are your hosts, Steve and Chris.
And good evening, cyberspace. Welcome to Squatch-D TV for today's date, August 30th, 2020. I'm your host, your guide, the Squatch detective, Steve Kulls, along with the man down there, Mr. Chris Bennett.
Hey, Steve. Man, we're live from a construction area, work in progress here, but hopefully I can step around the hammers and nails and stuff and paint and breathe the paint fumes long enough to stay awake during the show.
Yep. And, you know, I'm looking at this and, you know, if you need be, we can put some of our people in the chat room to work for you. So
I appreciate the help, all the help I can get. Yeah.
So I believe we have Dr. Hart on. Dr. Hart, how are you tonight?
I'm fine, and I can hear you now. I don't know what the problem was before, but it looks good. I'm on my cell phone, though.
Okay.
I could never get it on the computer.
All righty. Well, Internet Gremlins be. Now, I'm wondering if we're streaming on Facebook at all, because so if one of my if you could go over to Facebook real quick and see if we're streaming on Facebook, because it seems like we're only streaming on YouTube tonight. So if there's anybody on Facebook, say hello real quick.
Yeah.
So you're not sure what the issue is
there's
there.
B's on YouTube. Hi,
yeah,
B.
we got B and Frank who's saying hi
Frank.
to B. And we got Tack. And Frank saying hello to Tack. And so I don't know. Any Facebookers, say hello real quick just so we know we're there. But a heck of a show we have lined up for you tonight. We'll pull a graphic up. There it is. We'll get that up there. And we got this handsome fellow down there on the bottom, Dr. Hart.
Dr. Hart, welcome.
Well, thank you very much. I'm pleased to be here.
So, no stream on Facebook. Oh. So it looks like the gremlins are at work tough tonight.
Oh, man.
So anyway, yeah. Yeah, they'll have to catch it
All
on replay.
Yeah.
That's all right. Anyway, so we've been out for a few weeks. Some, life happens, and it's been kind of a rough week. Chris is moving into a new location, and I haven't been feeling the swellest. I'm feeling wonderful now for the last week. So everything is good. So I don't know if one of you guys can either Tack or Walt, if you could go over to my Facebook page and just post a comment that the Facebook stream is not working.
Yeah.
And if you want to pop over to YouTube, that would be awesome. So anyway,
B says that the Facebook's not working, Steve.
Yeah. No, I can't add it, Mike, because I'm using Restream. And you can only use Restream, and that's it. So there's really not much I can do about that. Yeah. If I'm doing, 3 separate feeds, I could do that, but I'm using Restream because we're also broadcasting to the websites as well. So nope, unfortunately, when you use Restream, you can't go back and. So anyhow, we'll get right to it. Chris, for the longest time I've been a real big critic of the Ketchum study, aka the Sasquatch genome project, and the reasons. And I've said for a long time that if they're all getting the same results, it's either contamination or the primer is wrong, and because I believe she's patented the primer. So, but
like somebody's chasing dollar signs when they start doing stuff like that. Rather than do something to further science, they do something that furthers their bank account, and that's not good, you know.
And Dr. Hart is, to me, he is the expert on this whole thing, because from the science side, we can get into the whole. We published it. We've published it. We're published in a de novo journal, which turned out to be owned by her, and we can talk about all that kind of stuff. But tonight, I really want to talk about the soup and nuts of the study itself. And Dr. Hart's written this great book on Kindle. It's called the Sasquatch Genome Project: A Failed Study, and there's a forward in it by Dr. Jeff Meldrum. So of course we all know who Dr. Meldrum is. No surprise there. So, Dr. Hart, I'll let you go into a preamble, why and, why'd you write the book, and, the kind of an overview of what you think happened to this study.
Okay. Well, in 2013, when the original paper came out, February 13th, 2013, I was aware that it was about to come out, so I went ahead and paid my $30 and bought the access on the website, on the journal website. And by the way, that's not unusual for journals to charge for that. So people who, disparage that, that's not a real issue. And I thought, well, gee, this is a great opportunity to learn a little more about DNA and identification of unknown species. And so I took this on as a personal growth project. And since they had all the sequences downloadable from the website, I went ahead and downloaded the nuclear DNA sequences of the 3 samples. Then I went and tried to find a database that had some search engine with it that I could make some comparisons of these DNA sequences to known species. And I found it. It was actually, it's free. It's part of the National Library of Medicine, as part of the National Center for Biotechnology Information, and people submit their sequences from journal articles and elsewhere. And so it's got, I don't know, it might have 1 million species or more by now. And millions and millions of sequences, short sequences, long ones, total genomes, everything. So I thought, wow, this is great, and I just have to learn how to use it. So I thought, well, let's get going, and I did, and I did some preliminary searches, and at the time, I had to learn what is a significant hit. I mean, if you match one sequence to another, is 90% good enough? Is 95, 99? And so I was working this out, and I had a little misconception at first about how significant 95% was. And I had to learn that there are conserved genes which don't vary too much between species because they work very well and evolution has not changed them. And that was the problem here. By Ketchum using a human reference sequence and matching her sequence to that to sequence it, it was a bias toward things that have some conserved genes. And so anyway, I sorted through all this and I found some disturbing things. And one of them was that, she purportedly sequenced 3 whole genomes. Well, actually, she only attempted chromosome 11. That's one out of, your 23 different 24 different chromosomes. And so there's something wrong there. But then even in chromosome 11, her sequences were only a small fraction, 2% and less of the total chromosome. So that plus the fact that 2 of these sequences, 26 and 140, they only overlapped 31 by 8%. So these 3 samples are not from the same species. And so that's kind of disturbing to begin with. And they're not like close to the same, that is, to primates or something. So the more I got into this, I started to look at the mitochondrial, and there were anomalies there too. There were too many extra mutations for some of those samples, way beyond the human normal range. And so the more I got into it, I thought, well, gee, there's some basic things here that are wrong. And I tried to publish a paper, which at first was unsuccessful. Then I did eventually publish 2 more on various aspects of this. And so last year, 6 years after the original paper, what prompted me to write a book was Scott Carpenter came out with a book, and it was all about conspiracy theories and how mainstream science had treated Melba Ketchum so bad. And he made the statement that nobody has ever critically reviewed her work and come up with scientifically based criticism, which of course was not true because I'd been doing it for 6 years and he knew that. So I thought, well, look, it's time to put all my results together in one book where people can compare them, look at them, get the background information, not have to go everywhere on the internet. So this has taken me about 10 months, and it would have taken longer except for, unfortunately, the COVID virus, which gave me a lot more time to do things.
Right.
So that's the history of it. Let's put it that way.
So out of that, let me run by you a couple of questions. So it seems like to me, and I've always said this, it seemed like Ketchum was always trying to fit the evidence to her hypothesis rather than the other way around, fitting the hypothesis to the evidence. And that does seem to be the case here.
Well, it's okay to have a hypothesis going into one of these things. You know, they teach kids in grade school that you have a hypothesis and then you test it and this and that. It's okay, but when you bias your results by trying to compare your sequence with and sequencing it based on a human reference. That's not appropriate for an unknown species. You need to do, what they call de novo sequencing, where you don't assume anything. You use these short fragments. You look for overlaps and build bigger ones that way, and then overlaps between them, and build your sequence that way, not making any assumptions.
Right.
So it was a preconceived notion.
So
and
that would basically mean why, when she turns around and says, well, all the other labs I've sent this to has gotten the same results, because she's sending them the same primer.
Well, yeah, I'm not quite sure what she means by that. If you use a human primer and you have a contaminated sample, you're going to get human sequences, even though they may be a small fraction of the total amount of DNA, because the rest won't prime if you use a specific primer.
Gotcha.
And, by sending stuff to Family Tree DNA, they're one of these ancestry companies. They're a good company.
Mm-hmm.
Their sole modus operandi is to take your buccal swabs and use human primers and give you a sequence and a haplogroup and tell you who you're related to. They have no methodology for unknown animals. And I proved it. I have friends, and they gave me cat DNA from her mouth, dog DNA, and another lady, her horse's mouth swab. And I sent them to Family Tree DNA, and they all came back failure to amplify, failure to sequence. So, their methods will not do other animals.
Gotcha.
And you can send something that's contaminated with human, but 99% something else, and you're going to get the human result, because that's all they prime it for.
Gotcha. So that's pretty.
That's not appropriate, for an unknown.
And the fact that she was only using chromosome 11, so she wasn't doing the standard. She was just using one chromosome, correct?
Well, yeah, here's the thing, of course, the effort goes up with however many different chromosomes you want to try to match this to, or to try to do a whole genome. I don't know why she picked chromosome 11. It was never justified or discussed in the paper or elsewhere. It did not, she didn't get much of chromosome 11. In these 3 samples, one of them was about 2% of chromosome 11, and one was less than 0.5% of it. So even that didn't work too well. That should have been a clue. If you have the right reference sequence, like a human, and you got a human sample, you should be able to sequence a lot more than just a couple percent. And so, that's a clue too. But
and was Scott Carpenter aware that you were, doing this stuff before he came out with his publication saying that there's been no serious?
In my book, I have an appendix with a letter that was sent to him and Melba Ketchum, and he and I have had a couple short exchanges based on a couple of things on his blog, and I think he took my response down. And I've been talking about him on sites that he's aware of and involved with, so he knew what I was doing. I don't know why he would say that nobody had ever critiqued that work scientifically, because he knew damn well that I had, but it doesn't fit his paradigm of what happened here.
Right. Yeah, to me, it sounds there's a lot of disingenuousness. And just to get a little sense of humor out of this whole thing, one of our fans here, Frank, who's in here every week, Frank, and we're good to hear you, Frank. Good to see you're not working tonight. Says Melba has a great joke for breaking ice at the party. What do you get if you cross a banana and a human and a lemur, a Bigfoot? So
Well, Dr. Hart, it seems to me like if you have 2% of chromosome 11, that would be difficult to make a determination of a species, wouldn't it?
Well, that's still a lot of base pairs. The biggest one, the sample 26, had 2.7 million bases in the sequence that was determined. Sample 140 was 2.1 million, and sample 31, which came out pretty much human contaminated with fungus, that one had 531,000 bases. All those numbers are plenty good to identify a species. You know, that's enough of a sequence, but you would have expected much more from the methodology she used. And the reason she didn't get it is because these two animals, number 26, the bear, number 140, the dog, are sufficiently far from human in evolution that trying to sequence with a human reference sequence only gives you those genes that are conserved, that haven't changed much over mammalian evolution. So you don't get the whole picture because most of the genes are very different and they won't align with the reference. So you see what I mean? It's trying to put a square peg in a round hole kind of thing.
Right. She was getting like gene sequences that would have create something with 4 limbs and a single head and stuff like that, and that pretty much swatches over to about everything in the animal kingdom, including humans.
Yeah, that's a simple way to put it. That's true. There are a lot of genes. I mean, after all these common mammals, they have a heart, they have lungs, they have a brain, a spinal cord. They walk on feet, 2 or 4, depending. So, you know, and their biochemistry can be similar in some cases. And so that's why you get some of these genes that are nearly the same. But 95% is not good enough when you're dealing with these genes. You need over 99%, or preferably 99.5%, to identify a species with conserved genes. And I don't think that team, the Sasquatch Genome Project, realized that. I didn't know that. You're not born knowing this. I'm a physical chemist. I had to learn it, you know. But after working with this for years, since 2013, you know, you can figure some of this out.
Yeah, it took me a bit, but I kind of worked some of it. Like, okay, I'm no scientist, so looking at it, you know, I'm more of a forensic guy. I'm an investigator by trade. So looking at some of it, I could see where, not 100%, but where, hey, something seems wrong here. And it seems like you have these mutations or because of contamination, and she's calling it Sasquatch in every single case. And her, you know, her justification always was, well, it's human DNA and this quote unquote angel DNA. And I've heard ruminations of that since 2012, even before the report came out. Yeah.
I don't get into angels. You know, that's a belief thing. It's not science. And, you know, that these are extraterrestrials and all that. I can't deal with that. I just try to use known scientific methodology or develop something close to it. But so that's where I stand on that. But there were many of the tests that she reported in her paper that came out so anomalous. Her answer was always, it's an unknown species. But really, the more logical explanation in these cases is that she was either using the wrong primers or that it was contaminated, or both. Or degraded, too. Degraded is another problem with this. All of these issues I brought up in separate chapters with individual searches and matches to databases and figures and philo trees. That's a tree of evolution where you show the relationship between species based on their DNA. And so I'm hoping people will get into it a little deeper than some of the emails and so forth that, you see typically on the internet.
So there's been some questions where they can get your book, and I got the link actually scrolling in the banner below.
Yeah, cool.
So,
well, one thing, and this wasn't the case at first, but if you go to Amazon.com and just look up my name,
yep,
the book comes up, and it's now available in paperback as well. At first, this is kind of interesting. I put in Haskell Hart, and I got Haskell Har, Harr only, and he's a famous drummer who's written a lot of drum symphonies and books to learn how to drum and stuff like that. I couldn't get through it, and finally, I think they put me in the database or something, and it'll come right up. There's only one book. I've never written a book before, so there's only one.
Gotcha. There is a question in the chat. I'll put that up on the screen and saying, what does Dr. Hart think about Dr. Meldrum's work?
Well, Dr. Meldrum is an expert in the evolution of bipedal motion in primates. That's his very specialty. He's an anthropologist and a biologist. And I think his work on footprints is solid. Unfortunately, it's not enough for mainstream science to accept the new species. But his analysis of footprints and the type of foot that would have to make those and everything seems sound to me. Now, I'm not an expert in this, so I should tell you that I'm just looking at it as another scientist, and it looks pretty good to me. It's not enough, unfortunately, to prove, and I don't even think DNA would necessarily be enough to have it recognized. I think you're going to need a body somewhere, probably. But I have a lot of respect for Dr. Meldrum, and I asked him to write a foreword, or if he would, and he said he would. So I wouldn't have put him in my book if I didn't respect his scientific abilities, Nobody's perfect, though. I will say that, not me, not him, not you guys, not Melba Ketchum. So, there can be things that people do that, they have to retract or that aren't quite correct.
A couple of quick notes. Not Yankee. Our good buddy Nottingham Yankee from across the pond, which is Nottingham
Welcome.
Yankee.
Yeah.
Welcome. Mick, our good friend Mick's got a question. What are the chances that Sasquatch DNA shares enough similarities that it could easily misidentified as human? And I think in a DNA study that would definitely show up the difference. But
well, this is a very good question, because I guess if you put a gun to my head and said, what is it closest to, I'd say it's probably closer to human than anything else.
Right.
I may be wrong. That's just based on some very sketchy evidence. But if you have enough sequences, there will be places where humans will differ greatly from chimpanzees, although overall it's only a few percent. But there are places where the differences are more extreme. And we can hope that there would be enough differences from a modern human that they could be picked out. But they may not be so different. They may be just a slight mutant of a human, or something of that nature. So we don't know, but and I don't know what the odds are if you're asking for a percentage, but I think there's a good chance that it would be difficult to tell the difference, or you would need a very long sequence or a series of fairly long sequences to really prove that this is not human. Because it could only be, a few tenths of a percent different or less.
Right. But do you have any doubt that Sasquatch is a primate?
Well, I think it's chapter 19 in my book. I actually saw one, and people think I'm probably a skeptic and all that. And, I'm scientifically skeptical, but I saw one in clear daylight for
Wow.
about 2 seconds or a little more. And I had somebody go out there later, and it was way bigger than this 6-foot tall friend. And it was broader, and it was red and hairy. And I heard it break a branch or a stick as it walked.
Yeah.
And so, and my distance was a little off. I thought it was 50 yards away, but when we went back and measured it, it was 30 yards away.
Yeah.
But I've been known to overshoot the golf green, so that explains that. But anyway, 30 yards away, and so I believe it exists. I don't know exactly what it is, but this was 11:20 in the morning, and on this particular group, we weren't drinking or anything, and we don't do drugs and stuff. And
Right.
I'm a pretty good observer of nature, and I've had better than normal vision when I was younger and so on. And I picked this up out of the corner of my eye and then turned, and there it was. And it was surrounded by bright green foliage, and it was in an opening. And so the contrast couldn't have been better. This is not one of those light gray on dark gray out of focus things at all.
Right.
Unfortunately, I didn't get a picture. I don't know that I could have in 2 seconds. People say, well, why don't you shoot it? Well, I wasn't prepared for that, and I'm not sure I want to shoot one now. They may be too close to human for me. And, I couldn't have pulled it off.
I'm sure it took you a couple of seconds to figure out what the heck you were looking at. You're probably like, what?
Well, I'll tell you what, we were out there looking for them, and, I didn't expect to see one. This is one of my first expeditions. And, I was checking some string traps where you tie a string between 2 trees, and if it's high enough and it's broken, well, maybe you got one coming through there, or it could have been an owl or a bird or something. But I was checking these, and, I hadn't seen anything in the previous 2 days, 2 or 3 days. So I was a little unprepared, but, I wasn't like, I didn't have to think very long about it.
Right. I always tell people, because the psychology end of things and how people behave and how creatures behave is kind of my thing. And I've always said, people always say, well, why didn't you shoot it? Or why didn't you take a picture of it? Because it takes that for the first few seconds, your mind is trying to process what you're seeing. You're not like what? Really? And then by the time you're thinking, oh, I got this, it's gone.
Yeah. I've never been able to focus my camera that quickly. I mean, you can get a fixed focal length camera, but it won't magnify very much, and so on. And a couple of seconds is not long.
Oh, right. One of the more aggravating things is lay people who turn around and say, well, with all these camera phones and all these, electronic, why don't we have more pictures? Well, the fact is we probably do have some, but they're blurry because, in a forest, that camera will focus on the first thing it sees instead of what you want it to shoot. So I've said that time and time again. And a couple of comments out there, and we have another question, a couple of more questions coming up. First is Shane Corson, our good friend out there. It says, I've got a lot of respect for Dr. Hart. I'm glad you have him on the show. And
well, I'd like to say thank you. And I was on his show a couple years back.
Shane's an awesome guy.
I think we lost Dr. Hart.
So we'll continue with the questions, but this has been a fascinating discussion, and I'm sure he'll just pop back in. So that happens with the phones occasionally.
It is. Yep.
But
well, I am actually thrilled that we have a scientist that has actually had the privilege of witnessing one of these things, and that is so great, man. So great.
The last scientist that I think what's the biologist up in I want to say he's either Washington or oh God, he's been on a few TV shows. I haven't seen him in a while though. He's been in
the red-haired guy.
No. He's a black-haired guy. Black-haired guy, mustache. Actually had an encounter while he was in a tent. He was a field biologist. Oh, there he is.
Sorry,
There we are.
I hit
okay.
a button.
We're used to it. So there we go. I don't know how to fix it. I'm trying to rearrange. We now can rearrange the tiles.
I see.
So now we got you at the top.
Well, normally during the show, I'm always looking from the left to the right because I'm checking out the chat and then looking at you guys. Now I'm having to do this too. So it's not only watching a ping pong game, I'm also watching a basketball bounce or something.
All right. Question from E-Man512 from Texas. He says, how about Sierra Sounds? Do you have any opinions on those, if you've heard them or not? Ron Moorhead's Sierra Sounds.
Yeah. I bought the recordings and I guess that was several years ago, and listened to them. They do sound unique. They don't sound like any other animals I've ever heard. And that's about all I can say. They could be the real thing. They may well be. They sure don't sound like anything else I've heard out in the woods, day or night.
We were talking while you were off the air about another scientist who had a sighting, because it's always refreshing to have a scientist who actually has seen one. And I was trying to think of the biologist's name, and thanks to Shane, it's John Moisinski.
Thanks, Shane.
Yep, that is exactly who I'm talking about, Shane. I knew you would come up with that. So Mick says, considering that we can't be 100% positive that we have a specimen of Sasquatch DNA, it would be difficult that against the human DNA. Okay, that wasn't a question, but okay. Tack, our good friend Mike. So could you explain, the good doctor explain what he saw in the face of the creature, what makes him lean towards human, just out of curiosity?
Well, actually, my own personal sighting, I did not see the face. It was angled away at 45 degrees, heading kind of diagonally away and looking the other way. But of course, I've looked at the Patterson-Gimlin film many times, and that, I guess it's not just the face that makes me think that, but something that's walking on 2 feet. I mean, none of the apes do that for very long distances.
Right.
And so smart as to avoid us at will and only get in contact when it wants to. And I do think I've seen a couple of things, short DNA sequences. They seem to be close to human, but they weren't long enough to really make a positive call on that.
Gotcha.
I could be very wrong. It's just kind of putting a bunch of little things together. They're all kind of circumstantial, though. Right.
Yeah. Yeah. I mean, it is amazing. We have so much circumstantial evidence. Now, I know, Chris, you probably want to ask this question, but I'm going to cut you off at the pass. I'm not talking about eye shine or ear flutter.
Okay. Right.
But do you think with the advent of eDNA, do you think that's going to help, eDNA collection? Do you think that's going to help us in our quest if we can get a hold of that technology?
Well, I have some pretty high hopes for that, actually. I guess you're familiar with the Gimel study, from New Zealand, where he went to Loch Ness, and they sampled it different depths, different times of the year, all around the lake. I think a couple hundred samples altogether. And they found DNA from every class of vertebrates: fish, reptiles, amphibians, mammals, and birds were all in the water. Some of which are not marine animals. They're terrestrial animals, and their DNA obviously washed in from streams and so on. And they found dozens of species, and the closest thing to a Loch Ness monster was the eel. But European eels typically only go to a couple of feet long. But some fish, have gigantic exceptions. And it could be a large eel. They found no new species per se. It was just a lot of eel DNA there, but it was known that there were eels in there. Of course, you have salmon and, other fish too.
Yep.
But I thought that, and I'm working up a proposal. I'm trying to work up something that somebody with a laboratory would take seriously, and collecting stream water in a watershed in different locations that would pretty well cover the drainage, and do the same technique, extracting it from the water.
Right.
Now, that would make a lot of sense.
It does have other benefits in that if you're trying to get a feel for all the animals in an area, and you have anecdotal sightings, this and that, but you might discover some species you didn't know were there. So it's a good baseline for an ecological study of the species that are present, and maybe even you could standardize it somehow, whether they're going up or down. And, for example, something like the red wolf is thought to be possibly even extinct due to hybridization with coyotes.
Mm-hmm.
But, you might find some red wolf DNA in something like this. In the South, it's a more southern animal. So I think there's possibility. It's a long shot, just depends on what streams they pissed in, I guess, something like that, or whatever. Took a bath in. It's what you get are cells that slough off from an individual. It can be fecal matter, it can be urine, it could be just skin, this washing, blown off hair, and then the DNA is extracted from that. But it's going to be degraded.
Yeah, I would think that. I would think that Washington, and particularly Texas, and some of the watershed areas that you guys have, there's a lot of sightings that they're along the water, in the water, grabbing either fish or other type of edible sea plants or creatures, or not sea, but water plants or creatures.
Yes, actually, in East Texas, there's a couple of rivers that there've been a lot of sightings along. Let's see, which one is it? The San Jacinto River, and let's see, I'm trying to think of the other one. I'll think of it in a minute. It goes through Lake Livingston, and then east in the Sabine River.
Yeah. Yep.
Right.
Yeah, they like to be near water, and I can understand why. But I think it has promise.
I think it's very interesting, especially, like all the waterways that connect they go down from Oklahoma down through East Texas and into Arkansas to the Fouke area, those whole tributaries. I find that very fascinating that they seem to travel down there. And I remember going into Fouke, Arkansas. It's one of the funniest things. I went into the Monster Mart, and there was a bunch of guys talking there, and they saw my hat, and they were like, "Oh, you're not going to find them around this time. We got a drought. We have no sightings when there's a drought." Just like as a matter of fact. Yeah, it was like, wow, well, that's, interesting that they would notice that. And again, it's very true to how primates behave. It's like, okay, there's no water. They're going to move on to another place.
Yeah, and you have the Sulphur River going through that area, and the Red River, and I'm trying to think there's another one too, but yeah, that's a promising place to collect some samples, I think.
Absolutely. Mick asks, what's Dr. Hart's opinion on different types of DNA collection kits? I don't know if that's too broad.
Well, this I don't have a laboratory, and I've never done a laboratory sequencing experiment myself. And so I would defer to people, some of the professionals, the contract labs or academics who actually do this in the lab for your answer. I can't give you a good answer. There are water sampling systems, different kinds of filters. Some of them pump the water through the filter. Some of them you collect the water in a jar or something and pour it through a filter in a vacuum flask. And so if you're talking about eDNA, there's some possibilities there I've seen. You can look around on the internet and find them probably easily. As far as other test kits, well, basically you want to wipe, if it's something like blood or saliva or something, you want to wipe it off with a wet Q-tip, and that will dissolve it more. Then put it in alcohol and or freeze it, and that'll preserve it. But there's all kind of variations. There's some stuff that helps preserve it that you can add right away, some lysing agents, some the surfactants, and so on. It's kind of like what they send you with the kits from these labs that do your DNA for you. They have a solution. You stick the Q-tip in, and so. But yeah, try to find somebody who's actually doing this in the lab. I've read about these, but I can't really compare them.
Tag asks if there's a lab that you recommend for DNA testing, because he's sitting on some samples of hair. And I have seen those hairs too. Very interesting.
Well, I'll tell you what, I looked around a little bit, and the lab that Brian Sykes, Professor Sykes at Oxford used, is in Pennsylvania, I believe it is. And they have published some papers. Terry Melton is the contact there. And she published a paper with some others where they took old forensic hair samples where the case is over and the cops gave them to them just to see what they can do. And they were able to get mitochondrial DNA out of little snips of hair less than 1 centimeter long.
Wow.
Cool.
And so I think they know how to do this. They also sequenced his 30 samples that Sykes had in his study, right. One of which was one of Ketchum's, and it turned out to be the black bear, just like I thought. And there was no contamination. They have a method. They wash in alcohol, not water. I'm sorry. They wash in water, not alcohol. And they ultrasonicate these hair samples and get all the external DNA off of them. And then the good stuff is on the inside and doesn't come out till you do another process of breaking it up. And so they know what they're doing. And I talked to her, and I think for the 12S RNA gene that they sequenced for Sykes, I think she was quoting me in the $1,300 range. So it's affordable if you really want to do it. I'll tell you what you should do first, though, and I've come into this later, but looking at these in the transmission optical microscope, the so-called compound biological microscope, can really eliminate a lot of other common animals. If Ketchum had done this competently, they did it, but they had some guy that didn't recognize it. I recognized it as immediately a black bear, the hair picture in her paper. It's unmistakable, and you can eliminate a deer, a bear. They're quite different. And primates have very little medulla in the center, and it's interrupted. It's not continuous. and it's very thin compared to the overall diameter. And, at least you can say it looks like a primate before you spend money on sequencing.
Right.
And you can get one of these microscopes. Mine costs less than $300 before tax, and then I got accessories, so I maybe spent $400. And it's just, I would not do anything with a hair without looking at it first.
Heck, now they even have like the $100 ones you plug into a computer.
Yeah.
And,
That's right.
and you can
bought
take a
my
picture of it.
granddaughter
And if you have any doubts, you can take a picture of it and send it to somebody like
That's
Dr.
right.
Hart or Dr. Meldrum or Kathy Strain is another source I use,
Well,
to say,
Yeah, the best one out there on the coast that I know is in British Columbia. It's Cindy Dosen,
okay.
who was
Yeah.
part of the Olympic project.
Yes. Yeah.
She's done the most that I know of anybody. She's much more experienced than I am. She writes you a whole report about it and everything. So you can get her attention. Lately, people say that's difficult, but anyway, if you're doing hair, you got to look at it first.
Yeah.
Otherwise, you could be wasting your time. You cannot tell with the naked eye the difference of some of these things, because even humans have different diameter hairs depending on where in the body it came from and which individual, what the race was, and so on. So you can't even identify a human one by looking at it for sure.
Well, I always found the interesting thing is that I've seen hair, and I remember getting a hair out of a stump, and I remember looking at the hair, and I just pulled out the magnifying glass, and I could see it was a red hair with a black tip, and I automatically knew that this is not a primate hair because they don't, delineate like that unless they're dyeing their hair. But it was very clear. To me, it looked like it was probably a fox hair, and I was like, okay,
that would be one possibility.
Yeah, but it definitely wasn't a primate like hair. It was too short. It was more like a fur than a hair sampling from a fur coat rather than that. And I've seen some stuff like, I'm kind of puzzled because, there was even reports that like the whole FBI thing with Peter Byrne sending the hair in, and it turned out to be deer.
Yeah.
It was like, why didn't they catch that before they even sent it? To me,
that's right. They didn't look under a microscope. You have to look in a microscope because that with the transmission microscope, you will see the medulla in the center. And by adjusting the intensity of the light and possibly even using a dark field lens, you will bring out the details of that medulla. And that is the most distinguishing feature of the hair, the medulla. The next would be the scales on the outside, the follicle. And so these are easy to do. You can make an image of that outside in just nail polish remover, and use I guess it's colorless nail polish. You just stick the hair on it, and when it dries, you rip it off, and it leaves an image of the outside of the hair. It's very simple.
Wow. Okay. Cool.
Wow.
Man, well, as always, I say,
learned this in the last 3 or 4 years, and I got enough experience collecting samples that I won't be fooled by a black bear. That's for sure.
One of the other things too that as somebody who has experience in forensic collect, and I do. I mean, I've been an investigator. I've had to forensically collect different pieces of evidence here and there. And it started with my brother was a police officer who was actually a fingerprint expert. He was the guy who did all the printing. And now I've learned that they get DNA from fingerprints.
Yes.
And I'm wondering that if you take a cast and a fresh cast anyway, and do a quick eDNA off of it, if we would get any DNA off a footprint, or we would get any DNA off a fingerprint. People claim that, there's a handprint on the side of a house or on the glass or on the car or whatever. I'm wondering if we can get DNA off of that before we even print it.
Yes,
Well,
I know that they have done that with human fingerprints, and the techniques these days are so good at amplifying the DNA to get many, many billions of more copies in just a couple of hours. They keep duplicating it by amplification procedure, and so you don't need too much, and especially if you don't have a lot of contaminants, it can give you plenty to sequence, and that's
So
one thing. I'll tell you what, in a footprint though, rather than mix it with all your plaster or whatever you're using, I would just collect the dirt and
wow.
extract
Well,
it. And this is what Meldrum was doing up there in Washington State with those nests,
right.
And they never found anything that was promising, from what I've heard. I haven't seen anything written, but you may know more about it.
I wonder, it kind of makes me wonder whatever happened to that nail, the board with the nail that got stepped on
Okay.
back in Vancouver Island. Remember that?
Well, the nail that I recall was in Ontario.
Yeah, was it?
At Snellgrove Lake.
That's it, not Vancouver, Snellgrove Lake. Yeah.
Yeah. Well, I'll tell you, the latest I've heard on that, I looked around a bit, and it was sent to a guy at the University of Minnesota, I think it was.
Sounds like Dr. Kurt Nelson.
Yes, that's him.
Yep.
Actually, it went to Disatel first, and he found that there was something interfering with his analysis. And Nelson was able to figure that out. It was the iron and zinc in the galvanized nail that had leached out and interfered with the sequencing the amplification reactions. So he was able to eliminate that, and he got something. I don't remember how long his sequence was. It wasn't very long, but it had like one unusual mutation that is also found in chimpanzees. But
wow.
I think he even agreed that there wasn't enough sequence, a long enough sequence to draw any conclusion. Because you can have a random mutation, and just one isn't going to prove it. You need a long sequence with several of these differences that you can point to.
And like I said, we have like the greatest audience. So we have some questions and some comments too that are coming in that you can comment on that.
Great.
E-Man 512 TX says, thanks for answering my questions. Some of the earlier questions. Does the doctor think there's a cover up going on like the guy from How to Hunt thinks? And as you know, there's this guy by the name of Steve Isdale out there, How to Hunt, who has been spewing the embracing the Ketchum study and the Patterson massacre theory. And he's saying there's this huge cover up and that we're all part of this cover up, which is kind of ridiculous. But
yeah. Well, Melba Ketchum said I'm either a paid government agent or a quack. And that's her view. She thinks that we're all involved in a conspiracy because that, we conspired.
You're a government. You're a government agent supposedly trying to knock down her study, but yet you're admitting to seeing a Bigfoot.
Yeah. Okay. Well, I'm glad I put the chapter in the book there just to let everybody know I'm trying to be as objective as possible here, but perfect, And so anyway, I think that's just a convenient way to dismiss the scientific evidence and focus. That's what Scott Carpenter did in his book. It's all about conspiracies and people trying to keep her work from being published and the Nature reviewers. By the way, I've gone in detail through the Nature peer reviews and the other journal peer reviews. And by the way, I didn't know about them until after I had done a lot of my work. And I agree with every one of their 16 points. And I came to the same conclusions independently.
Wow.
So
there you go.
I think from my point of view, they were fair and they were justified and scientific about it. And that's another chapter in the book you might be interested in. So, I think conspiracies are easy to charge and hard to prove, including all the famous ones about Kennedy's death and everything, It's easy to come up with something, but hard to prove.
Well, I think it's a cop-out, truthfully. It's a cop-out. Oh, well, it's a conspiracy.
Sure.
Yeah. But I kind of funny. She's calling you the quack, but she's the one who's saying that Bigfoot's braiding her horse's hair.
Okay.
Well,
yeah.
I really, to be honest, I had high hopes for the Melba Ketchum study when it was first being talked about and she was working on it and it was in the works. And I was like, oh, finally, we've got somebody that's going to do the work. And then after it came out, there was a lot of stuff in there that talks about religious icons and things. I mean, what was it? The giants from the Bible or something. And
oh yeah, Nephilim, whatever you call them. Yeah.
Nephilim.
That's it. And I'm like, wait a minute, that's not science. She's just dived off into opinion right there. And there's no way that she could get that from the DNA results, because I don't think we had the Nephilim DNA sequence on file anywhere that I know of.
Yeah. Well, that's true. That's very true. And of course, we don't have any reference sequence like all the other species in these databases.
Right.
But nevertheless, if you have an unknown species and you have a long enough sequence and you compare it to enough other species, you can construct this phylo tree that shows where that species lines up in the tree of evolution. And if it's deeply embedded in the primates, you can be sure it's a primate. It's not a dog or a cat or something else. And this is what I found with her samples, number 26. It was deeply embedded in the bears, just where a black bear should be. Right. And the other one, the dog, the same thing. And I've got these phylo trees that compare them to other carnivores, and it's pretty tight. You can't argue with it, because not only does it compare with, say, the bears in the case of sample 26, but it also has the correct relationship to all these other species for a bear to have, So that's the way to do it. Her phylo trees, well, one of them had primates, that's fine. And another one had fish. It had a carp, 25 species of bony fish, and a couple of dozen sharks, and a chicken, and a mouse, and a human. I mean, that's not a phylo tree of primates, Right. And those are not the primates' nearest relatives. And the other one had nothing but mice and humans. I mean, of all the primates that it should be related to, nothing, just a human and a mouse. So they didn't know how to do phylo trees, and their data wouldn't have supported their thesis, as I showed. And the other sample, number 31, was human, and I believe that's the one that shows the more primate phylo tree, even though she has it identified as another sample. So it's such a mess.
Like I said, our audience is rocking tonight, so we got a ton of more questions.
Okay, I'll try to talk less and answer the questions.
No, that's fine. That makes my job easier.
Okay.
All right. So from Shane Corson, what are Dr. Hart's thoughts on DNA work on the collection of ticks in an area where a sighting has occurred, or an area where something substantial has occurred in regards to a Sasquatch? Good question.
Yeah, I'm not as familiar with that work. I have heard of it generally, that of course they eat, they suck blood, and in that blood would be DNA of the species that bit. But I don't know how long it lasts. Whether you got to get a really fresh
Tick, yeah.
sample from a bug that just bit something, how long it lasts in their body, because I'm sure it degrades eventually. It's a possibility. I mean, the whole Jurassic Park thing was based on mosquito blood or
mosquito.
something. Yeah, mosquito. That's fiction, but it's not out of the realm of possibility. I don't know that much about it though.
Dr. Hart, you must have been reading my mind, because I was sitting here thinking about, I was going to tell Shane, yeah, man, you remember that movie where they made the dinosaurs from the blood they found in mosquitoes?
Yeah. The ancient mosquitoes, yeah. That is kind of interesting.
Maybe we could make a Sasquatch.
Now, Mick, our good friend Mick, he's going to complicate things right now.
Uh-oh.
Can Dr. Hart comment on the possibility of the existence of a dog man, considering that canine and hominid DNA are incompatible? Well, I don't think that's so.
Yeah, well, they're pretty different. I found that out just by doing some comparisons. Dog man, I don't know. Melba thinks it exists. She said one of them raped one of her horses.
Oh, jeez.
So you better talk to her on that one. I don't know much about that.
And who's calling who the cat the quack?
It seems a bit far-fetched, but I don't know.
Mix also says my ex's meatloaf is proof of random mutations.
Oh boy.
Yeah, Mick is ragging on the ex's meatloaf again. I don't think meatloaf is supposed to glow in the dark.
Mick and the meatloaf.
Yeah, Mick. My ex-wife was such a bad cook. I bought her a pressure cooker. We used to have to eat off the ceiling. Anyway, this question's from Media Palace. Does the doctor think that Todd Standing is a hoaxer?
Yes, I do. I don't follow that too much, but when it came out with, I guess he took Meldrum on one of his expeditions and kind of convinced him. But overall, I think a lot of that is hoaxed.
Yep.
The pictures are just too close to some other costumes and things.
They don't seem genuine. One of the things
according to the evidence, anyway, it's all this stuff is hoaxing.
Yeah. I mean, if you look at some of the stuff, it's pretty. I mean, you got 3 headshots and all 3 look different. I don't know. I mean, I think Sasquatch are dimorphic, as all primates are, but I don't think they're that quite dimorphic.
We're not going to talk about the gorilla hands tonight, are we?
No gorilla hands.
Okay.
And
another time.
And Ken Gerhard commented on that. He's a showman. Hey, Ken. Wow. So, Doc, can you just remind our audience what is needed in your basic 101 DNA collection kit? I'm out there doing research. What should I be carrying in a DNA kit? Just for the basics. Right.
Well, as far as protecting it from contamination, of course, you need some gloves. A mask is good. And I would even, if you're really serious and worried about hair, you could put on one of these doctor's caps, kind of keep your hair out of the picture.
I don't think I have a problem with that.
Yeah. Well, that's great. I'm losing mine pretty bad too. But here, it depends on kind of sample you're looking for, You just need to use forensic techniques, and they're pretty well described around in literature. But then what you do next can make a difference too. You need to preserve the sample either in ethanol, which is ethyl alcohol, and or freeze it. and get it to the laboratory, and then the experts in sequencing will know what to do from there. But it is important because the stuff will degrade on a day scale basis, you know. So preserving it immediately is important, and you know, just use some sterile containers and gloves and stuff, and you know, it just depends on the sample, but
Yep.
those are the basics. DNA is insoluble in alcohol, so that'll just preserve. It kills all the bugs and stuff, so that it won't get degraded. Now, interestingly, Melba Ketchum used alcohol to wash her hair samples, and that's not good because it won't dissolve the external DNA. It's water that's the best for that. And I think she used water and alcohol, but I sure wouldn't wash with alcohol. That's for preserving it.
Gotcha. Now, one of the things too is that, you know, like I said, if somebody comes across like a blood on a leaf or a branch, I always tell people, don't try to collect the blood off the branch. Snip the branch and collect the whole branch. And that would be a lot easier than or you get blood on a leaf, or say dirt. Collect the area. If it's a solid piece like a stone, collect the whole stone. Don't try to transfer the blood off the stone. Take the whole stone. And that way, you know, you have a lot of sample there if it's
yeah.
And
well, the thing is, if you can get it to somebody who's experienced in getting it off of whatever substrate you have, it's better that way to, you know, not fumble in the field with something you're not as familiar with.
Yep.
And how to dissolve the blood, but not get all the other crap with it. And yeah, cutting off a leaf and sticking it in one of these vials and immersing it in alcohol would be the safest thing.
Yep.
And let somebody who knows how to do this do the tricky part.
And please, please, please do not put your DNA evidence in a plastic bag, because the humidity and that will kill it. So always paper.
That's not good enough. No. And so those are just basic things. I mean, you can read about this elsewhere and get a little more detail.
So E-Man asks if you've heard of the Area X or the Brushy Creek area in Central Texas.
Brushy Creek sounds familiar, but I swear I can't recall any details there. What part of Texas is it? Probably East Texas, huh?
He said Central. Looks like Central.
Oh, Central. Oh. Well, I live in Central Texas, more or less, South Central. I'd like to know where that is. No, I haven't really.
And there's no two. Shelley Covington Montana sells DNA collection kits for researchers. So you can, if anybody's on the book, you can reach out to her and check it out.
Yeah, she's done a lot of research on how to do this, and that's right. I've seen the kits, and they're very complete, and that'll get you started. Yeah. Yeah.
Well, you know, and like I say, DNA collection, all you need is one of the things I always tell people too. If you anytime you collect, also, and this is from an investigator standpoint, is, and like I say, when we're in the field, we're not the scientists. Our job as investigators and researching this stuff, we need to think about what we do. I've always said that, you know, as an investigator, I collect evidence to give to the prosecutors or the attorneys or whatever to go to court. And my job in this field is to collect the evidence and give it to the scientists to prove what they need to prove or disprove what they need to prove. So, you know, keep yourself out of the bias of it. Let the chips fall where they may. But at the very least, some gloves. I don't think masks are hard to find now with all this COVID stuff. I mean, maybe, you know, maybe a year ago, like, yeah, okay, I got to buy a mask. Now everybody's got them pretty much. So you have a mask, and in some cases you have multiple, you know, so take a mask, take a pair of nitrile gloves and a paper bag, and you have everything you need to collect a piece of evidence. Basically, take it, put it in the bag, log it, picture it before you take it from all angles. Tack made a great point earlier. If you have one of these 3D scanning programs on your phones, and you get a track and you want to collect a dirt for DNA collection, scan the print very carefully so you have a 3D picture of the print. Collect your dirt into the bag, you're good to go. So all of that, it can be done very easily, very cheaply, not overcomplicated. And that's what we need to practice on. Too many times we're gawking at things too much. You got to treat it like a crime scene. Keep everybody out. Only one person should go in. Make sure you're properly PPE'd for it, or wearing your personal protective equipment. Go in, collect it, bag it, tag it, and log it, and then you pass it off to the next person.
One of the things is, the cost of having that DNA sample processed is expensive, and it's been my hope that as the science advances, it'll get cheaper.
Now, I don't know if Dr. Hart knows this. In very early portions of, and this is when I started having some of the concerns about the Ketchum study. How much was Dr. Ketchum charging for the DNA tests on the stuff? Anybody know? It was $2,500.
Really?
And she would have the person,
submitters?
Huh?
She charged the submitters that amount?
Correct.
I did not know that.
Yep. And they would have to sign this draconian agreement that they will be sent the results of the tests, but they don't own the results of the test.
Yeah.
That they can't talk about the results of the test.
Yeah.
That basically everything was shredded. So I'm paying you to do this, but you're telling me, okay, if it's Bigfoot or not, but I can't say if it's Bigfoot or not. So to me, the whole thing was quite draconian. And believe it or not, I had gotten that retainer from JC Johnson when he was alive, because he was part of that project for a while.
Right. Now, I didn't know she charged the submitters. I do know that, she was also funded by Wally Hersom.
Well, he was paying for some of the submitters. Yep.
Yeah, I did not know that. Okay. And that's a pretty hefty price. I mean, I don't have enough other examples to say whether it's overpriced or not, but you can get a commercial analysis for probably a little less than that. But you gotta know a little bit about this so you can ask the right questions and make sure they're going to do the right thing.
Yeah. Well, I always thought, at least back then, 10 years ago, when at the start of all this, I thought, okay, 2,500 is not that bad. I mean, I think that was like the going rate. It was around that target price. So that didn't seem too high for me. Excuse me. But the fact that she would charge people submitting this for her study, to me, that just, it doesn't ring right. You're gonna take my sample, you're gonna call it your own, or you're gonna call it mine, but in the study, but you're gonna turn around and claim the results as your own.
Well, a commercial lab gives you total rights to the results to do whatever you want with them.
Right.
And I think they'll even agree not to share them with anybody else. So it's a completely reversed situation. I'm surprised because she got a lot of funding from Wally Hersom and I think Adrian Erickson too, but that she would even be charging people. But I'm not, again, when I think about it, I guess I'm not so surprised.
Hey, so while we're on it, what do you think of the Matilda photos?
Well, they look fake to me, but again, I'm not an expert. I think somebody like Bill Munns, who wrote a book about the Patterson Gimlin film, and he's a Hollywood makeup and costume artist, and analyzes the motions of the animal in the Patterson Gimlin film and compares them with what you could do with a costume. somebody like that would have to speak. And I think he has, and I forgot what his conclusion was. I don't know. It just looks too much like some costumes that the people have made,
I'm actually.
But I can't, I'm not an expert in that. I can't really be definitive. That's just my feeling. I just don't believe it.
Yeah, that was with Munn's put up as, partly in the Matilda photos that to him it looked like a Chewbacca costume, which when you start including those into the study, that really I mean, I remember one remark that was made about the study from one of the peer reviews, and it said that the first like 5 minutes or the first 5 pages were all on stuff that wasn't necessary, the Matilda photo and this and that, and they were really, it was like she was almost trying to pitch the existence of a Sasquatch before the evidence came out.
Sometimes an introduction will take you a little bit far afield just to put it in proper perspective.
Right.
But it should. I thought she went a little too far, and that video of I could never see the head on that thing. The creature that moved on the ground that was sleeping.
Oh,
it's hard to believe. If you got that close to it, why didn't you wait till it woke up and get a picture of its face?
Yep.
Yeah.
Or something.
Yeah.
Or pull some hair off it or something.
So here is the big topic of the whole thing too, and something we kind of steered away, but we want to get back to it. And we got about 26 minutes left to go. So let me pop this up. The Justin Smeha Bigfoot steak.
Yeah, that's her sample 26, the one I showed to be a black bear.
Right.
And she did nuclear DNA on it and also mitochondrial DNA. And the mitochondrial was kind of a mixture of 2 different human haplogroups, I believe, from what I could tell. And one of them was Justin Smeha's haplogroup, and then the other one was different. But it had a couple of unique mutations that are not found in humans together, and very rarely even one of them. And they are found more often in primates. And I have a chapter on that too. I think it's about 16 on unusual extra mitochondrial DNA mutations. And so that sample, though, the bulk of it was a bear. It could have been, well, here's the scenario. The Sasquatch was killed by the shooting, but it went some distance and wasn't found. Then 2 bears fought over it, getting the DNA all over them. And in the process, one of them tore a bit of flesh off the other one. And that's what Smeha found, because the bear DNA showed it was more than one bear. Bears come in contact with one another just like people. And so there are indications in what they call the microsatellite loci that there's more than one bear in that sample too. And so, you can imagine a bunch of things, but there are some unusual human mutations too, a couple. And so it could be contaminated with Sasquatch, but there's not enough evidence to prove that.
Gotcha. So, when Dr. Sykes said that, he considered that sample was a bear. Was that?
He only looked at one hair.
Okay.
And the hair, I'm sure, was a bear.
Actually, he got the bloody boots.
Yeah.
But I don't think he was able to extract DNA from that. From what I heard, that was unsuccessful. I've heard another version, but I've heard him say twice, I think, that they couldn't get it was on a BBC program, and he's sitting down with Justin Smeha. He said, unfortunately, I couldn't get any amplifiable DNA.
Right.
So what
Okay,
he
gotcha.
analyzed in his paper, though, was a hair.
Right. That came
That
from the
was
snake.
true.
Yeah, I know a lot of our audience here. We had interviewed Sneha probably a year after the event with the driver, and somehow the story mutated a little bit since his appearance on Bigfoot Bounty, where he actually choked the one out instead of it dying in his arms like he first claimed. Then he said he choked it out, and so there's a lot of people that do not believe his story.
That's right.
And
Well,
yeah.
according to Scott Carpenter's reproduction of the polygraph test, he passed 40 different questions on this subject, and 2 of them I've got in my book, and they relate to whether or not that sample that he found was the exact same sample that was cut and sent to Melba Ketchum and to Tyler Huggins and Bart Cucino, who did independent analyses on it. And he passed that, but of course, polygraph isn't 100%. They say it's about 89% accurate.
And there was a big question about the polygraph examiner herself too. She fell under a lot of controversy as well.
Oh, really?
She had actually took an online course to become a polygraph examiner. And she's using something, I guess, polygraph in a box. That was the software. So it was a little, there was a little, questioning about, and, Ken Gerhard made a good point too. A sociopath can also pass a polygraph test.
Yeah, some people are good at it. It is a skill that some people can have.
Right.
And I don't know. Yeah, you can't really say for sure whether he was telling the truth or not. But anyway, it's information.
Yeah. Uh-oh.
So who else we got? More questions?
Joni, I think you ought to get this book by Dr. Hart. You'll understand scientifically why it's not real. Anyway.
Well, the evidence is real, but her conclusions about the evidence.
Yeah. The evidence is really came from like the hair JC, submitted came from a pipe, inside a pipe. There was some blood or something inside a pipe. But yeah, I mean, gets so, people will follow, people like, it turned out to me that it seemed like, it seems like that people like Melba, have created these stories around this study to gain this cult following. And, a cult following is exactly that, a cult. I don't think that any one of her rock solid supporters that are standing next to her today are going to change their minds by any means. Neither will any of the juggernauts, such as, a Standing or a Rick Dyer or any of those folks. If you're going to stand next to those person and espouse their stuff, it's not steeped in science. And that's why I had Dr. Hart on today. So, here's a man with no agenda except, let me take a independent look at this. And he came out the exact same results as the rest of the people who peer reviewed it, all the same problems.
Right.
I don't know how many times that if you sit there and say the sky is green and 5,000 people turn around and say, no, the sky is blue, and you insist the sky is green, it's time to get a better filter. That's all I can suggest to people. Because in no way, I've looked at it. I've seen, why is everything about the Ketchum study mired in controversy? There wasn't with the Sykes case. There wasn't with the Sykes book. There's not here. It's just everything is straightforward here. It's amazing too how every sample that Melba had was a Sasquatch. Was there any that she excluded from the study that said that it wasn't a Sasquatch? Do you know that, Dr. Hart, off the top of your head?
Well, it's very interesting. Her table number 1 has all the samples that were presumably analyzed in the study. And unfortunately, a good number of them never appeared in the paper anywhere. So I don't know what happened there. And, if you go out in the woods and collect 111 hair samples just randomly over many states and even Canada, would you really expect them all to come out as a Sasquatch?
Yeah.
Don't you think some might be something else, like a bear,
Right. Yeah.
a deer, a bison, a rat, a fox, or whatever? It isn't very probable. If they were that common, we'd have probably solved the problem a long time ago. If there were that many of them,
Yeah.
that just doesn't add up. And
Yeah.
there are many other things that I address in the book. It's kind of hard to explain them all here without using tables and figures, but
Sure.
I am open for questions and answers. I've reinstituted my membership in the Bigfoot forum, so there's a link, a topic on my book, and people are invited to ask questions or challenge anything. I welcome that. So have at it.
Yeah. Yeah. Exactly. Yeah. Well, I think I want to thank you for coming on tonight, Dr. Hart. It was awesome. I know we got kind of a late start, but the information you put forth today, pound for pound, great information, and definitely gives us a clearer picture of what was wrong with the Ketchum study. And again, folks out there, it's now available in print edition as well, not just Kindle edition. Go to Amazon.com, Google Haskell V Hart. Remember the heart, because and Haskell V. That way it doesn't get mixed up with anybody else. The Sasquatch Genome Project, a failed DNA study. If you even Google that, it'll probably come up with the Amazon thing. So go out there and grab that. That's going to be a great read. And what I love about books about scientists too, Dr. Hart, is they make great reference.
Yeah,
they make great reference. I mean, I keep all the, I got Dr. Krantz's books, Dr. Meldrum's book. And, if I need a reference, I go to those books and maybe with the exception of Ivan Sanderson, because he has a lot of earlier dated stuff.
Yeah. Well, I will say this, that I think every research group, if you have a group of people that go out together and stuff, somebody ought to be familiar with looking at hair under a microscope.
Yep.
And identifying species. You can buy samples, or I pick them up off the roadkills, I get a whole bunch of different animals right around my house. And so, and another person ought to be familiar with some of these DNA issues, which I address in the conclusion and throughout the book. Not everybody needs to be up on that, but if you're going to submit samples, you need to know a little bit about this. Otherwise, you might not get back what you thought you paid for. And I'm not saying there's any intent to deceive, but I mean, if you don't at least talk the scientist's language a little bit, it's very hard to be sure that he understands exactly what you're trying, because there are different ways to analyze these samples with different primers, different goals, different objectives. And so somebody needs to know a little about that. And I hope the book would help.
Right. Yeah. What about the mid-tarsal break? Well, that wrong doctor, Adam. Wrong doctor.
Yeah.
We Dr. Meldrum. And I meant to mention another good book to reference, and books that are written good to reference is by this guy too, Ken Gerhard. So
yeah.
And
the last one was a great Bigfoot overall covering of the subject and the sightings and things. Yeah, I agree. I have it. He gave me one.
Joni is still trying to make her argument, I think, because she says, P.S. I've talked to Melba. My condolences. I've talked to her too. And I have one quote. Fairy knots. Look that term up. Look what it does to horses' hairs. Devil's knots, fairies' knots, witches' knots.
Well, we're not saying that Melba is a bad person or anything. It's just that the evidence that she put forth, her conclusions were wrong.
But as a scientist, Chris, as a scientist, she should be no better and be more objective.
Well,
that's my
point. That's the thing. Okay, when she was first started, I was one of the followers. I was like, all right, Melba's going to do a great job. And then, but when she got to the point when this religious stuff came out, that was the edge of a cliff. Okay, she went off this cliff, and I'm standing there looking at the edge of the cliff, and I decided I'm going to back away because there's no way that you can have biblical stuff come from DNA. Okay, I'm sorry,
Chris. This stuff goes way beyond that, though. Between the Bigfoot's braiding her horse's hair because she woke up one morning and all the hair was knotted. That's because there's such a thing as fairy knots, devil's knots, witches' knots. They're not. Now we got a kooky story out there. Somebody said it braided their American flag. Oh my Lord.
I saw that. Yeah.
but let's go back to Melba all of a sudden losing consciousness, waking up with her pants unbuckled, and now claiming she's had some sort of an experience. She believes a Sasquatch did it to her in her mind or whatever. Where I mean, that's just some of the background noise. Let's go back to the beginning. Here she's claiming she got published, and then she was outed by journalists saying that she owned the publication, and that was the only. So of course she's claiming she made a claim and she got caught on it.
Yeah,
she got caught on it. To me, I
got published on my website too. Anytime I want to update it.
To me, and then we go into the stuff like Scott Carpenter does, saying, "Oh, nobody ever looked at this." Some scientific. Well, if that's not true, not only did Dr. Hart do it, but what about the people that peer reviewed it?
Right.
Okay, and we run automatically to it's a conspiracy. Yes, I'm sure Dr. Hart, myself, and a bunch of other people got together and said, "We're going to tear down this study." Well, what would the purpose of that be?
Men in Black.
If you have a conspiracy theory, it's got to have a legitimate conclusion. There's, you look at the JFK conspiracy. That at least has kind of a legitimate conclusion that this was either done by mob or the government or, the Cubans or whatever. At least that has some logical conclusion that they're working towards. Throwing out an abstract such as this is a conspiracy amongst Bigfoot researchers and scientists to shoot down the Ketchum study. Well, there's no conclusion. Why would they do that? Well, and then they get into, it's jealousy. Oh, hand me another one. Hand me another one. The fact is it's egotism on the other side. Is that here's the study, I'm putting it up for peer review, and when people poo-pooed it, when the scientists looked at it and said, no, it doesn't stand, she insisted like an impetulant child and went out and got her own little journal and then tried to make a claim and got caught. And then, to me, it just, nice person. I don't know.
You can't claim that Dr. Hart is a Men in Black government agent sit here to make everybody believe that there is no Bigfoot, because he's like, I've seen one.
I'm still waiting for my check.
Well, and he's not sitting there with dark sunglasses. He doesn't have a white shirt and tie on,
right?
And he doesn't have the thing up here where look into this. We're gonna make it.
Well, I was disappointed at what I found. I was hoping that her study would solve this mystery for once and for all. And in the process, I'd learned some things about DNA and sequences, matching and identifications. And I did learn that, but I was disappointed. I had no skin in this game. I came into it with an open mind, and I still have an open mind, and any of my claims in the book can be checked. I mean, I give enough information that if somebody wants to, they can go and use this BLAST program against a government database and see what they find, you know. And I led somebody through this over the phone, in fact.
You know, it's just, to me, it's just amazing. And the people that support Melba, well, I'm not saying you're bad, but please put a better filter on. You know, if you're out for the truth, if you're out for science, you know, put a better filter on, because Melba turned out being laughed at. You know, you look at her press conference and, you know, oh, she's having this big press conference and she stands up and she spends 15 minutes, you know, trying to dress down the scientific community, trying to dress down other Bigfoot researchers, saying how they're jealous and this and that and the other thing. Sorry, you know, that's not the way to win a crowd over. You know,
right. The thing about science is, you know, once you have your results and if they're verifiable, that's all, nothing else need be said, you know.
But, you know, I said this very early on. If you look at the interviews with Richard Stubstad, he was now deceased. He passed away before the study came out, but he was part of the original gang of three, which was Melba Ketchum, Richard Stubstad, and very well-known Biscardi associate Bob Schmalzbach. Wow. And when that business got dissolved, Stubstad started to speak out, and Stubstad specifically spoke out about a lot of this is ego-driven. A lot of this is, you know, ego-driven. And that was the biggest thing. She wanted the name. She wanted the marquee. Fine, she should have it. But she was all about being very, it seemed to me, very paranoid with the draconian agreements to process the samples, that it was about ego. And of course, what would you expect? I mean, it seems to me it was about ego, because here's all these peer reviews and she's jumping up and down saying me. So, and then now we have to build our cult following up by claiming this was going on and that was going on and that was going on. So to me, it's very clear what's going on there. So that's why she's in my Hall of Shame.
Yeah, I noticed. Well, I hope after people read my book, you won't put me in there too. But I really think I would just like to have some people understand this, and I'm not going to make any money on the book. It's priced reasonably, and you know that's not the reason. I just want the truth to come out, and I would appreciate comments, pro and con, polite, constructive. Give me everything you got, and because you know science evolves. Nobody's got all the answers. Nothing's the final word, really. So maybe I overlooked something.
And you know what? Nobody goes in that Hall of Shame for being wrong. They go into the Hall of Shame for not only being wrong, but being proven wrong, and then insisting that they're right and putting out false information. And that's exactly what's going, because after 7-8 years of this now, you know, just give it up. Melba, give it up. It's over with. Nobody believes it.
It's hard to admit after you put so much in and been on the TV and the radio and everything that you were wrong. But a big person has to do that.
Yeah. Well. Yeah. So anyway, folks, Dr. Hart, thanks a million for coming on. It's been a lot of fun. I hope you've had fun. And
I enjoyed it. And you guys are good conversationalists, and your audience had excellent questions. And as I said, I'll answer any more in detail if you go to the Bigfoot forum.
There you go.
So good to hear from y'all. And I'll try to continue to provide some answers if I can, or direct you to somebody who knows more.
Amen.
Awesome.
So anyway, folks, on behalf of Chris, any final words?
I just want to say thanks again, Dr. Hart. We really appreciate it. It was an honor having you. And I'd like to thank our audience for our great audience for being here with us once again. And remind you, if you're on YouTube, watching on YouTube, please hit subscribe, share, comment, whatever you got to do. It helps us out with the ratings. Helps we get more stuff. We can put more links in there. So thanks, everybody. See you next time.
Absolutely. And don't forget to subscribe, like, and share to our YouTube page. Every little bit helps. Hit that notification bell. Learn when we cut new videos and new posts. So anyway, folks, on behalf of me and Chris and Dr. Hart, we want to wish everybody a great, wonderful weekend or week ahead. Be safe, keep healthy, God bless, and of course, keep on squatching, folks. We'll catch you all here next week, Sunday, 9 p.m. Eastern, Squatch-D TV.
Good night.
Hey folks, you've been watching Squatch-D TV. Join us each week, Sunday night at 9 p.m. Eastern, for the latest on the Bigfoot mystery. As always, we thank you for being our loyal viewers and encourage all to subscribe to our YouTube page at youtube.com/Steve Kulls. As always, have a great week. Stay safe. God bless and keep watching.